Right. Okay, perfect. Yeah, this is the latest map of the spread of corona right now, and there have been three main strategies to contain the spread of the virus. One would be social isolation, travel restrictions, but we don't really know how long this is still going to take, and it has become quite a burden at this point. Then vaccination, same thing. We still don't know how fast this will be. And then last but not least, and I think this is a very important strategy where I believe a lot of opportunities lie to contain the virus, and this is what I'm going to be talking today. So basically, a year ago, there was one issue about these testing strategies, we were detecting corona based on these diagnostic or clinical tests, which very quickly run out, we had shortages. One of the tasks of a lot of universities and research centers were to develop in-house PCR testing strategies. And same, the JKU did that, and my team took up the challenge to develop these PCR tests at the JKU. And our aim was basically to develop a test that was quite flexible, meaning that we could tap in different reagents so that we would be able to overcome this reagent shortage. And also we would have a test that would be hands-on, meaning that we wouldn't require expensive robotics and yet stay high throughput, meaning being able to process 180 to 300 samples a day. I know these numbers sound ridiculous at this point, 1 year later, but at that time, compared to a robot or the clinic that could process maybe 600 samples a day, it was actually quite a good quite a good number. At that time, we did not know either what we were getting into. We thought it was just a lab exercise, since we have the expertise of my team is in PCR. But very quickly we realized that there's a series of logistics behind this testing, and we were part of the task force also together with the Gesundheitsbundesministerium to try to integrate this testing with in all its different steps. Basically, we were trying first to set up the probes and of course the method, the PCR itself in the lab. Then we very quickly realized that there were also shortages in the sampling. So we also were part of developing different test kits and the stability of samples in these test kits. And last but not least, we were trying to integrate our testing pipeline together with all the steps from inputting data to validation, reporting, and communicating the results to the Bundesministerium as valid results. Also, as you might know, all these, because of this very complicated logistic behind this testing, sometimes tests would take up to 3 days to get a result. I didn't have to do much about the process itself, but also getting things into the lab, taking the samples, and all the way to the reporting. And I have to say, at that time, I was working, or we were working very tightly with the Kepler Uniklinikum, the COOK, with Elisabeth Schreier-Lechner and Christian Paar, who were actually doing the testing at the clinic, and they actually helped us quite a bit to try to integrate all these steps. Because of political issues, we didn't go all the way, but this is just an overview of all the things that are involved. Okay, so let's go to the science. So currently there's 3 tests available that are used for detecting corona. The PCR-based test usually focus on the genetic material of the virus. It's a molecule called RNA. For those who don't know, RNA is a product of the DNA. This is the basis, detecting these RNA is the basis of this PCR. for those who have not heard what PCR is, PCR stands for polymerase chain reaction. This is absolutely not relevant for this talk, but maybe it might become important to win the next trivia game. So you can forget it right away if you don't need it. But so that I just have said it. Then the antigen tests are based on detecting also viruses, but now the protein, it's based on detecting the protein on the virus. Most of these antigen tests are developed to detect the spike protein, which is the main antigen on the CoV2 virus or the coronavirus. And last, we have these antibody tests that actually detect whether a patient or an infected person has developed antibodies against the virus. All right, so I will continue talking about the PCR-based test. Just a quick overview how the sampling is happening. You all know that, or maybe have experienced that the sample is being taken via this nasal throat swab. So this swab is inserted in your nose. It's quite uncomfortable, but then this swab is placed into sample buffer that keeps the sample intact, or also just keeps the sample stable, and then it's brought into the lab and processed by what we call a thermocycler or a PCR machine. Similarly, antigen tests don't require a big machine, so you can do it on site. So you also need to have this nasal throat swab put into a buffer, lysis buffer that releases the virus from the cells, and then you have these strips, chromatographic strips that work like a pregnancy test that tells you presence or absence of an infection. Similarly with antibody tests, where you just take a blood sample and add this to the test strip. All right, so going further into what this PCR test does. So as I mentioned beforehand, the PCR detects the genetic material of the virus. The virus is an RNA molecule that is a 29.7 thousand nucleotides long. It has different regions coding for different things. It's not relevant here. But what is relevant is to know how to design a specific and sensitive test. So here I'm showing the different mutations. The beautiful thing about corona is that now all this lingo is known by you guys, and you have heard at one point. So changes in the nucleotide sequence that have accumulated throughout the viral genome, and you can see that some areas have more mutations than others. So you don't want to place a PCR that in a region where you accumulate a lot of mutations, because sooner your PCR will not be able to detect this variant anymore because you have too many changes. But you also don't want to take something that is very conserved, because then you might detect other type of coronaviruses. And let me show you here what I mean by this. So in gray is, or what I show here in gray are what we call the primers, and in the middle are the probes, and this is what gives the test the sensitivity to pick up just the genetic material of SARS-CoV-2. It will not pick up SARS-CoV-2 of the 2003 strain because we have two differences right for the probe that will make this test unspecific, that will make this PCR not to work. And for example, compared to the MERS corona, we have many differences that will not get amplified. But of course, if you have a difference, a new mutant coming up, then this might also make your test not work, make that your PCR doesn't work anymore. So that's why you need to find a balance between conservation, but at the same time, uniqueness. So once we got that, and also other researchers found out the perfect place to set up your targets, we do what we call a PCR. So polymerase chain reaction is nothing more than getting copies and copies and copies. And this is an exponential process. And you monitor the making of copies of very unique targets by the machine called thermocycler that records the fluorescence or the light emission, and you can what we call an amplification plot. So a sample will give you an amplification plot that is exponential. A sample that has no virus will give you no amplification. And the PCR test is quite quantitative, meaning that if you add different amount of viruses, so going from a very high virus load, so 10 to the 7 virus per milliliter to 10 to the 3 or 1,000 copies per milliliter, you get a delay in the cycle number where you get a signal. And this is exactly what we call a CQ. So maybe for those who have been working with tests already or detecting viruses, the levels of viruses can be correlated with CQ values or are inversely correlated. As you can see, the fewer viruses you have, the later you start with the PCR. So 100 copies of virus per mil gives you a very late CQ versus high number of copies gives you an early CQ. And so the PCR test is very quantitative, and you can go back and forth, or you can always convert these CQs into number of viruses, which has been quite useful in this pandemic. The whole process is starting with the sampling. You need to get the sample into the lab, hopefully taken by some clinical persons, or nowadays even self-testing. The next step is to release the virus from the cells, and this is done usually during a cell lysis step. And then you want to also clean up this genetic material. It's called the RNA extraction, and this RNA extraction is one of the most laborious processes of the corona detection, and many PCR-based tests have been trying to get rid of this step. Then you, of course, well, not of course, but since the genetic material of the virus is RNA, which cannot really be PCR'd, as we call it, you first need to do a step called reverse transcription. And what it does is to convert this RNA into a DNA molecule such that you can then put it into your machine and do this PCR. So what we did in the lab was to combine these two steps into just one step that can be carried out within one step. So we kind of cut these things short and also cut the handling short and work out this RNA extraction such that it could be done with multi-channel and magnetics in a plate format, and this way speed up the process. Overall, we took about two hours to extract 88 samples. And the next phase was to validate our assay together with a diagnostic test. For that, we used samples that were measured in the cook of real patients, and we compared those measurements with our own measurements, called here JKU N1, JKU N2. These are two different targets that we are measuring. And as you can see, our measurements are quite in sync with the ones of the clinic, of the diagnostic tests. Which made us very happy. And we also started seeing that as you go up into very low viral loads, remember high CQ values means low virus levels, we start seeing some samples that did not render us an amplification curve. So this is exactly some of the issues of these tests, that as you get low in copy numbers, you sometimes miss to measure them, and we call these false negatives. But overall, we can say for those samples that have a higher viral load, 10 to the 4, 10 to the 5, we get 100% sensitivity, also 100% specificity. So all the negative samples were also measured with our JKU in-house test kit as negatives. All the positive samples were also measured as positives. And then we had the ones that have low viral load, and you can see that now our sensitivity drops down to 70% compared to 90% sensitivity of a diagnostic kit. But regardless of this difference, we were very, very happy to have this in-house test already ready to go in May. So thank you for my team that worked really hard on this, day and night and weekends to get this done. And we were ready to go in May. So Many things have happened in the testing world until then, and I want to discuss a little bit these and put these in contrast to our PCR-based tests. As you can see, so just to rephrase what these tests do, the PCR-based tests detect the levels of viral RNA, the antigen tests detect the levels of the viral protein, so both PCR and antigen tests detect the virus itself, versus the antibody test just monitor your immune response, meaning whether you have been infected by the virus or not. Then this is a comparison of the sensitivity of these tests. PCR is actually still considered the golden standard because it can detect the lowest virus levels so far compared to these other two tests. The antigen test only measures high numbers of virus, and the antibody test doesn't measure virus at all. It's just a post-infection measurement. So you can even you can have a positive antibody test but not have the virus anymore. So when are these tests being used or can be applied? Well, the PCR is fairly early on. The antigen test only can detect a positive in a very narrow window frame, versus the antibody test is post-infection. Of course, the time and the cost of a PCR test is still quite horrendous compared to the antigen test, which just takes minutes and is very short. So at this point, the antigen test is actually, because of this very short processing time and cost, and also because of it being that it can be done in-house and on-site, actually has been replacing this PCR-based test. Yet PCR-based tests are still considered the golden standard, as I mentioned, just because there's such a complicated pipeline behind it that ensures that the results are truly what they're supposed to be. So even though you get a positive antigen test, you have to go and do a PCR-based test for verification. So, I mean, ideally, we would have the two worlds together, having a test that is as sensitive as PCR, but as fast as an antigen test. But can we live with an antigen test at this point? Well, this depends. And this is now really what we're learning from these testing strategies, and where I think there's really a lot happening here. So here I'm showing how the viral, so here I'm showing on the x-axis the time of in days of starting from the viral infection onwards, and how the virus numbers start rising in your body. And as you can see, the infection starts early on, and you can start detecting an infection with PCR, with high sensitive PCRs early on, and only with the antigen tests within a very small window frame, because the antigen tests, remember, only detect a high virus load. Then we have a drop in virus because our immune system kicks in. So the question is, how sensitive do we have to go? Do we have to be able to detect positives or infections that are dropping in virus load, in viral load? And the antibody test just basically tell us whether we have been infected or not. So where this gray area is, where this point is actually where we can decide, is an antigen test good enough to contain the pandemic? Remember, testing is the idea of testing is pandemic containing. So now that we understand a little bit more about how the virus is transmitted and being spread, so basically most of the transmissions of the virus happen in a very, very small window where the virus is incredibly high, 10 to the 7, 10 to the 8. Meaning that 75% of the cases are coming from these super spreaders, or the big fish as they're being called. And yes, they're being caught by these antigen tests. So yes, corona PCR test is a golden standard. We can detect early infections by PCR, but PCR is still very limited. But it will be good to screen the pandemic. But the antigen tests are really good to find those super spreaders. So I hope, if you didn't understand much of the science behind this, at least you understood that it's really important to go testing, because it does make sense. And last but not least, I want to thank my wonderful team, especially Teresa and Maya, who really put a lot of work into developing this test in April and in May. And of course, the cook for it, and also the Rectorat for supporting me with this project. And you for listening, and I'm opening for questions.